Hi there, I'm a researcher at the University of New South Wales. I look after the Ramachodi Centre for Genomics, and this is a centre that serves the Sydney Basin and is involved with the sequencing of genomes. This ranges from bacterium all the way through to the sequencing of human genomes. And at that centre we also undertake a lot of analyses of gene expression in a field that's otherwise known as transcriptomics. Now, when you do large-scale molecular biology studies like genomics and proteomics and transcriptomics, there's a remarkable interconnectedness in the information. So you need genome sequences as the fundamental blueprint against which the proteomic and transcriptomic data are matched. And there's also a chance to use cross-correlation between proteomic and transcriptomic data to tell you which genes are ultimately made into proteins inside a cell. So while software exists to help make these types of comparisons possible, certainly at the start of this project there were some quite critical gaps. So for example, it was difficult to map peptides from proteomics directly against a genome sequence and to visualize that. It was also difficult to co-analyze next generation sequencing data and proteomics data, either to map gene splice sites or to help with the validation of newly sequenced and assembled genomes. So some of these analyses could be done manually, but it was really quite labour intensive and cumbersome to do so. So the aim of this project was really to create a software platform that would facilitate these different types of analyses. We call the project the Proteome-Genomic Nexus, or the PG Nexus, as it's all about building connections between data types and facilitate a series of visual and other quite in-depth analyses. Now, we're fortunate in that the Australian National Data Service, otherwise known as ANS, saw this as an important imperative, and we're in a position to fund this work. So through their support, we've built a software platform to do these complex cross-comparisons. But these tools are designed to be easy to use, and we expect they'll help many researchers to really co-explore genomic, proteomic and transcriptomics data. We've also established a means by which to feed genomics and proteomics metadata through to the data fabric that is established and run by the Australian National Data Service. And this will help make these large volumes of data which are being produced more discoverable. Now, in this video, two of the key researchers involved in the project, Dr. Ignatius Pang and Mr. Aidan Tay, will talk about the tools that we've developed. Then we'll have Professor Hazel Mitchell, an expert in diseases of the human gut, talk about how this tool has assisted aspects of her research work with Campylobacter concisus. We hope you find these tools as useful as we do. Thank you.
Hi, my name is Aidan, and I'm a researcher at the UNSW. Before the PG Nexus, there was no efficient means of integrating genomics data from next-generation sequencing with proteomics data from protein mass spectrometry. As part of the project, we have developed a series of tools. First tool is SAMA file, which converts peptides identified from mass spectrometry into a sequence alignment file. The sequence alignment file can then be used to co-visualize genomics, transcriptomics, and proteomics data in the Integrated Genomics Viewer, or IGV. By co-visualizing the data, users can intuitively assess the quality of the peptide hits and use this to validate genes and transcripts. We have also developed the Results Analyzer tool, which maps peptides matches from mass spectrometry searches to their genomic locations. The tool then generates a table, allowing users to filter and analyze the data using tools such as Excel or SQL databases. By filtering and analyzing the data, users can generate their own statistical reports of proteins and peptides depending on their precise application. We have also developed additional tools which are focused on bacterial analyses. The protein generator creates custom protein sequence databases based on predicted gene coordinates, or by translating fixed-sized overlapping segments known as virtual proteins. These sequence databases can then be queried, allowing project-specific mass spectrometry search results. The virtual protein merger uses the proteomics data from protein mass spectrometry to identify putative open reading frames from the virtual proteins. The mass spectrometry searches allow the virtual protein merger to recalculate the position of the start and stop sites, thereby providing peptide validation for newly discovered genes.
Hello, I'm Ignatius, a postdoctoral researcher at UNSW. With the PG Nexus pipeline, we can now co-visualize proteomics and transcriptomics data in the integrative genomics viewer to quickly validate genes and their genome annotation. We can also identify peptides that spans across alternatively spliced junctions. This allows us to characterize changes in alternative splicing in different types of human tissues and understand how they affect tissue growth. The above meets the challenges that are described by Professor Wilkins. The Semifire and Results Analyzer are now available in Galaxy, which is a web-based workflow application. By providing a user-friendly interface, it reduces the barrier to entry and hence quicker for users to learn how to use the tool. In collaboration with Intersect, we developed the web-based app to publish the input and results datasets and the relevant metadata associated with these datasets. This allows users to search the metadata and allows the data to be shared and findable by everyone.
I'd like to introduce you to Professor Hazel Mitchell. She's a professor in the School of Biotechnology and Biomolecular Sciences at the University of New South Wales. Hazel, can you tell us a little bit about your research with Campylobacter concisus?
Well, Campylobacter concisus was first associated with disease of the oral cavity, such as gingivitis and periodontitis. However, despite quite a long period of research, it still remains a contentious area as to whether it's involved with these diseases. There have been an increasing number of studies that have looked at the association between Campylobacter concisus and gastroenteritis, and many have shown an increased prevalence of this organism as compared with controls in patients with diarrheal disease. Our laboratory has recently shown that Campylobacter concisus is significantly increased in children with newly diagnosed Crohn's disease as compared to controls, which would suggest that this organism may be associated with both acute and chronic gastroenteritis. To investigate this issue, we recently sequenced and analyzed the genome of Campylobacter concisus strain isolated from a child with Crohn's disease. And this represented only the second Campylobacter concisus strain to be fully sequenced, and the previous strain being a strain from a patient with gastroenteritis. In addition, we have sequenced the genome of a concisus strain from a healthy patient. We hope to use the information from these genomes to be able to tell or to separate strains that may be associated with disease from those in healthy controls. Well, one of the major challenges was identifying the 1800 or so genes present within this organism. So gene prediction programs cannot reliably find all genes, and it was important for us to be able to find genes correctly before we could start any functional studies for these genes in my laboratory. These tools helped us to confirm more than 1,300 of the genes in the newly sequenced C. concisus genome, which was almost 3/4 of the genes we predicted to be present in the genome. Perhaps the most important thing that this work has helped us with is that we now have a remarkably detailed understanding of the genes and of the proteins of this bacterium. Armed with this information, we are now in a good position to determine through in vitro and in vivo studies which of the identified genes or proteins are directly involved in the manifestation of specific disease outcomes, including gastroenteritis and chronic infections like inflammatory bowel disease. This has the potential to allow us to identify genes that could be exploited as a therapeutic target for the treatment of Campylobacter concisus infections.